Hi, welcome to our poster. I'm Antonia and I'm presenting our new method for brain mapping called Neurolang for representing neuroanatomy with sulcus-specific queries. Our objective in this work is to identify and label subject-specific sets of sulci. To achieve this, Neurolang is a query-based mapping language which navigates the cortex according to spatial relationships to the primary sulci. Primary sulci are landmarks on the brain which are more or less stable. The other sulci are notoriously varied in their morphology, but what characterizes them are their locations on the cortex relative to primary sulci. A lot of what we refer to as tertiary sulci are often omitted from current atlases because they are small or shallow or unreliable, and they can often be grouped with their neighboring gyri, but many of these tertiary sulci are what create distinguishing features on both the individual and the population levels. In Neurolang, we have 36 sulcus-specific queries. To test these queries, we used the software MindBoggle to extract unlabeled folds from 52 Human Connectome project dataset subjects. We verified our secondary queries by matching the results of the queries with the maximum overlap with the Destriatlas sulcus, and we assessed our tertiary queries by their locations on the probability maps on the population level and the proportion of subjects in which a result was found. We can see from Figure 2 that some of the secondary queries were consistently matched with their Destriatlas counterpart, such as the inferior temporal sulcus, while some of them were not, or some of them had no corresponding Destrius sulcus. This can possibly be due to the fact that the unlabeled folds that we extracted had a much larger variability in their size and shape because we did not extract them using an anatomical atlas because we wanted to do the atlas labeling ourselves. Many tertiary sulci, such as the examples in Figure 3, were localized as expected, and they had proportion figures which were in line with what we expected from the literature. And in some cases, the tertiary queries yielded no results at all, which was also what we expected. So what we can gather from Figures 1 and 2 and 3 are that in each hemisphere we have a unique number of sulci as well as uniqueness in the shape and size of each hemisphere sulci. Therefore, the intent of Neurolang is to be used as a complement to current template-based methods. Neurolang can aid in the identification and labeling of such lesser-known sulci, which can contribute to the further understanding of structure-function relationships on the individual level, and on the population level, it can help us understand further the evolution of the human brain or comparative anatomy. Thank you for visiting our poster. If you have any questions or comments or would like further information, please do not hesitate to contact me. Thanks.